It's a pleasure to be here. What I'd like to do is fundamentally change healthcare in the next 10 to 20 years. And I've spent the last 50 years of my career figuring out how to do that. So I'm going to give a bit of a autobiographical view of some of the major events I've been involved with. And we'll end up, actually, one of the things I'll be talking about is a metric for measuring biological age, which is a proxy for aging. And we can talk about ways you can reverse that. We do know some things. We don't know it at the mechanistic level. We know it at the operational level. So when I went to Caltech in 1970 as an assistant professor, I was really interested in human biology. My lab was divided into two parts. I had some people working on technology development, and at that time, some working on molecular immunology. But what I was really interested in was human biology and disease. And in the sense of a biologist, anyway, what I was astounded at is the enormous complexity of humans. You've heard bits and pieces of it throughout this symposia. And what I really became convinced of is that the way we can begin to deal with human complexity is being able to make lots of measurements on human beings. And that set me off on a path that involved me with a series of paradigm changes. And I'll explain the first one in a little detail, and I could do for all the other six the same amount of detail, but the other six I'm going to go through very, very quickly. So the first paradigm change was, in a sense, bringing engineering to biology. And we did that basically over actually 40 years of my career by developing 6 different instruments that basically were the foundation for contemporary molecular biology. And I'll just say these allowed you to sequence and synthesize both DNA and proteins and do single molecule analysis. A really key part of our strategy was to recognize that academics could conceptualize new instruments and they could provide proof of principle, but the rest of it, making it robust, making it high throughput, had to be industrialized. So in all cases, we started companies that were successful in commercializing these instruments. And the other point that was really interesting is the first 4 instruments in an integrated fashion gave us the ability to do something very unique at that point in time. And that was sequence proteins at levels that were 200 times less frequent than had currently been analyzed with the protein sequencer, and then use the genetic code dictionary to convert that protein sequence to DNA sequence, and then use the DNA synthesizer to make a DNA probe. And you could use the techniques of molecular biology to clone that gene, and it could be sequenced by the DNA sequencer and so forth. So for the first time, we had access to proteins that were available in incredibly small quantities. And that really made it possible to do lots of exciting things. So for example, we sequenced a human platelet-derived growth factor and showed it was essentially identical to an avian oncogene, and that formulated the first oncogene hypothesis about viruses capturing genes that regulated growth and development and things like that. A really technically difficult problem was sequencing the prion protein. This is a protein that induces neurodegeneration in humans and mice. And the sequencing of that and the cloning of that gene opened up beautiful work Stan Prusner did that resulted in the Nobel Prize. We actually sequenced erythropoietin in conjunction with Amgen, and that turned out to be, after the cloning, the first billion-dollar drug, and so forth, a whole series of different things. And to give you an idea of how revolutionary this was, we had one issue of Science where we published back-to-back 4 papers that analyzed 4 different kinds of receptors that each went on to clone the genes and began to open up new fields. So I'm a big believer in this. New directions in science are launched by new tools much more often than by new concepts. New concepts just let you explore things you already know in new ways, whereas new tools open up the discovery of new things that have yet to be explained. So the second paradigm change, the automated sequencer got me invited to the first meeting ever held on the Human Genome Project in the spring of 1985. And 12 of us went and kind of passed judgment on whether or not it was possible and whether or not it was appropriate. We were split 6-6 on that latter point. And the general biological population was probably opposed at the 80% level initially on whether to do a human genome project, but we did. And of course, what the Genome Project gave us were 2 really powerful things. One, you could immediately obtain any gene that you wanted from the Genome Project by making primers and PCR amplification. And 2, from the point of view of medicine, really important, you could examine the genetic variability in human beings and correlate it either with wellness or with disease phenotype, and we'll return to that point in just a moment. The automated sequencer forced us to bring together really good chemistry, engineering, computer science, and molecular biology, and I started pushing the idea of let's have a biology department that's cross-disciplinary in nature, with that foundation being the fuel that could then take leading-edge biology and develop the tools and technologies and computational tools they needed. And at Caltech, that was an incredibly unpopular point of view with the biologists. The engineers, the chemists, even the physicists thought it was an interesting idea, but the biologists vetoed it for reasons that are obvious. So Bill Bates made it possible for me to go to the University of Washington and set up a department like that. In its 8 years of existence, it was really spectacular. We developed the key algorithms Phil Green did for assembling and quality control for the Human Genome Project. We developed the first 2 pioneering areas of the newly emerging field of proteomics, and we developed there the inkjet DNA synthesizer that Agilent still today uses to synthesize large amounts of DNA. But what happened is I wanted to build on top of this systems biology, and frankly, I found a big state university bureaucracy utterly impenetrable. So I resigned and in 2000 started the Institute for Systems Biology. And you've heard a little bit about what systems biology is. It takes a global and holistic point of view of things. It's all about biology is information. And of course, the fundamental units of information are DNA, RNA, protein, and a variety of small molecules. So we wanted to be able to analyze and measure those. And tools I talked about earlier were a beginning for doing that. The other point about biological information is it's enormously hierarchical. So information at the single molecule level, at the single cell level, at the single organ level, and the single organism level, each define units of activity of biology. And what is really key is to study at each of those levels individual molecules, cells, organs, or individuals, because averaging together the properties of these various populations smears out the most unique and important features. So N of 1 experiments are a central part of biology. And you heard very beautifully from the previous speaker about biological networks that mediate development, growth, aging, and similar kind of processes. And they become disease-perturbed. And if you compare disease-perturbed networks with their normal counterparts, you can get insights into biomarkers and possible drug targets and so forth. A really important thing systems biology did in the very beginning, the early 2000s, was to look at disease and define a series of the concepts that are central to how I think about disease anyway. So one was the idea of a systems approach to disease being systems medicine, where you try to understand the pathology and develop the technologies and computational tools you needed to carry out these studies. What we also became convinced of was that healthcare should be, in a global sense, predictive, preventive, personalized, and participatory, and we call this P4 healthcare. And in thinking about it, what became obvious at that time is that the healthcare is really two major domains, wellness and disease, and at that time it was 99.9% disease, frankly, still is largely today. But in 2014, we pioneered a study with 108 people where we did what we call deep phenotyping, that is making lots and lots of measurements on those biologically interesting molecules, and I'll define that in just a moment. And we call this scientific or quantitative wellness. And the pilot project I took to NIH and said, would you consider funding this? And Francis Collins said, NIH is only interested in disease. We aren't interested in wellness. So we used philanthropy to fund it, and then started a company called Harel, which generated 6,000 clients, each of which we generated longitudinal data clouds that, as you'll see, will really be transformational in just a few moments. So finally what happened is in the spring of 2016, I had the CEO of a local large healthcare system, Providence St. Joe's, come to me and say, would you like to become chief science officer, and would ISB like to become affiliated with us to help us with research? And we agreed to do that because I saw that as a way of bringing P4 medicine or 21st century medicine to healthcare. So what exactly is 21st century medicine? In the US anyway, I think there have been two big revolutions in the last 100 years in medicine. The first, in 1910, was a Flexner report that looked at 155 abysmal trade schools. And it made the recommendation, one, get rid of 2/3 of them, which they did. Two, science should drive the treatment of patients and the education of physicians, and Hopkins and a few other schools leaped on that, and together with the sciences of the 21st century, focused on disease, how to find it, and how to fix it. And of course, 21st century medicine is what you've heard about. It's systems medicine, it's the vision of P4 healthcare, and its manifestation, which is scientific wellness. And we'll show you more about that in just a few moments. But it was about the 4 Ps, but it was about understanding wellness, understanding disease, but especially understanding transitions from wellness to disease, because the initial transitions are where you want to reverse disease before it ever manifests itself as a disease phenotype. And of course, this gets a really interesting model for thinking about the continuity between wellness and disease and those early transition points. In several systems, we've demonstrated beautifully after one undergoes a transition, there's an exponential increase in disease-perturbed networks. So that argues compellingly that the transitions for disease should be right at that transition point. Today, medicine studies essentially only diseases that have already been diagnosed. So it's late-stage disease, and for many, they absolutely are irreversible. And of course, 21st century medicine is right about wellness and that transition point, as I've said before. So there were a series of converging things that came together again in 2014 that gave us a much clearer picture of elements that should be involved in P4 medicine. And you can see them listed there. They're exactly the kind of things: systems biology and scientific wellness and big data and analytics and digital health and things like that. But P4 is really different from contemporary medicine. It really is focused on being proactive. It's all about focusing on individuals, N of 1 experiments in medicine. It's about understanding wellness as well as disease, about this deep phenotyping and the creation of data clouds that will let you assess both wellness, disease, and the transitions. And as we heard yesterday, it looks at clinical trials and their fatal flaws. This is the slide that you saw yesterday. And of course, clinical trials now are carried out by, typically in cancer drugs anyway, 20,000 patients, half of which get a placebo, half of which get the drug, and then you abstract the data from those average populations. And of course, the fatal flaw there is the basic assumption in these trials is that the individual units are identical. And of course, humans differ genetically, and they differ environmentally from one another. They aren't identical. And here again, as you heard yesterday, here is the success rate of the top 10 selling drugs in the US today. The best rate is 1 in 4 works. The worst rate is 1 in 25 works. So it's just astounding that these kinds of things have been accepted. So for a P4 approach, what you do is have 20,000 individuals, each with their own unique longitudinal data cloud, and you could stratify those individuals according to key questions. For example, one thing we do know we can do in clinical trials is actually identify biomarkers that distinguish the responders from the non-responders. And that raises the really interesting possibility of clinical trials in the future, each being done with 50 subjects. 50 in the first case to identify the biomarkers that distinguish responders from non-responders, and 50 in the second case that are all responders. So you'll get a 98% or better response rate. And even with small numbers, the FDA has granted drugs on that particular basis. And as I said earlier, the two different domains, wellness and disease, are now slowly beginning to be distinguished one from another. And I'll argue over the next 10 to 15 years, the wellness industrial domain will far exceed in market cap the disease domain, as wellness and its early transitions do away with lots of chronic diseases and so forth. So again, the 108 pioneers, friends, that we persuaded to go through deep phenotyping to do a wellness project and to define exactly what quantitative or scientific wellness was all about. And the one key factor that really made us think seriously about how to design these experiments were a couple of independent studies on determinants of health in the US, where genetics contributed 30%, lifestyle, environment, 60%, and the healthcare system only 10%. And of course, these deep phenotyping gave us the ability to assess both genetics and environment and lifestyle. And what we did for these 108 were complete genome sequence analysis. Every 3 months, we analyzed roughly 1,200 analytes from the blood. So those are clinical chemistries, proteins, and metabolites. And likewise, every 3 months, we looked at the gut microbiome to quantify the species contained therein. And we had Fitbits and other devices to measure quantized cell features, blood pressure, pulse rate, activity, and things like that, generating data clouds that then led us in a unique fashion for each individual to a whole list of actionable possibilities that we garnered from the literature that, if acted upon by the individual, could either improve their wellness or let them ameliorate or avoid disease. And I'll tell you about my list. I was one of the pioneers in just a few moments. But what we did use were wellness coaches trained in nutrition and nursing and psychology that could deliver the actionable possibilities to individuals, because a big challenge is how do we get people to change their behavior? We had 70% compliance with the wellness coaches, so it was enormously successful. Here's the first blood clinical chemistry. So we had 91% with nutritional deficiencies, 68% with inflammation. And that wasn't inflammation from infection. That was general inflammation from diet and other things you heard about in the preceding lecture. 59 had cardiovascular difficulties and 56 diabetic. In fact, 53 of the 108 were pre-diabetics. We moved 8 to being completely normal, and most of the rest much further toward normal. And here's just a list to give you an idea of the actionable possibilities that I did. So I ended up losing 20 pounds, and I'm within 5 pounds of my college football weight. And that was done without real diet. It was done by modifying my diet, and it was done by increasing my exercise, which I just made much more intensive. I had 5 nutritional deficiencies that got corrected with supplements and vitamins. And vitamin D was really was really interesting, because 91 of the 108 individuals had low vitamin D, and some of us had really low vitamin D. 1,000 international units, in my case, didn't even budge my vitamin D level. And it turned out that there are 3 genes that have each 2 variants that block the uptake of vitamin D. If you have 2 or more of those genes, you need megadoses of vitamin D to get anywhere near normal. And I used 15,000 international units to bring myself up to normal, and I need about 5,000 international units to keep myself at that level. And for a lot of reasons, you don't want to have low vitamin D levels. I had high mercury from...
This experiment for who were the clients, the patients? This is him.
What? This is me.
For you?
Yeah. Yeah. I'm telling you what happened. I mean, and everybody had lists like this, but the lists were all unique. I'm just giving you an idea of what came out of this and why it made such a difference. So high mercury from tuna sushi. So now I eat salmon sushi, and my levels are down at normal. Inflammation, I corrected a lot of that with diet and so forth. I have ApoE4 gene, which predisposes you both to cardiovascular disease and Alzheimer's disease. So what I did is there's a beautiful continuous glucose monitoring disease where for 2 weeks you can do N of 1 experiments on diet, on fasting, on exercise, on all the parameters that you might want to know with regard to blood sugar. And I learned an enormous amount about what I should stay away from, because I have a gene that that predisposes me in that way. And one of the things that's worked beautifully is intermittent fasting. So, and the idea is you don't eat for 15 hours one time a day, and the easiest thing is to eat at 6 and then eat the next time at lunch. And it, again, made an interesting difference. But anyway, I think one of the lessons that I really did learn.
This improve methods, this fasting, intermittent fasting?
Intermittent fasting, it's called.
The health, or
It improves the health. Oh, yeah, yeah, yeah.
It's every day, or it's once in a while?
More is better, but most of us don't do it every day. So.
Every 15 hours.
Well, yeah, I mean, and you should do a long fast. So I've done a 48-hour fast. And actually, after you get by the first 10 or 12 hours, it's not bad at all. But anyway, so I think one of the things we realized is how important it is to be responsible and really push these things. And something I'm going to talk about in just a few moments is we now have a metric for measuring your biological age, that is, age your body says you are. And I'm 15 years younger than my chronologic age. And during the time I was in this program in Arafat, that I'll talk about in just a moment, I lost about 6 years of biological age. And the idea is the lower you are below your chronologic age, the better you are in aging. And so, but we'll talk about studies we've done there in just a moment. So here's a Fitbit analysis of my pulse rate, which is on the Y activity, and time is on the X activity. And it shows you how sensitive these things are, because this little blip is airplanes. So when you go up in airplanes and high, you're always under-oxygenated in most cases, at least I've been. This was the beginning of a flu that's the worst I had for about 10 years. And it took about a week to get the maximum. And you can see an enormous change in the pulse rate there. And then it went down to a more normal level and so forth. So there are obviously lots of digital measurements that you can make that let you get these kind of insights. But pulse and temperature are two that are very exacting.
This is very low level. This is low rate of pulse.
Yes, if you're in good shape, you can have really low rates. So my rate is about 40, 41, 42, something like that. So, and most people are probably 60, 55, something like that. But, and I started in Arafat. Mine was about 60, and it's come down that much. So these are all parameters that say you got somewhat healthier. So the idea here is that, as we heard earlier, what you want to do with scientific wellness is follow the actionable possibilities. And we're convinced that for many, they can actually take you up into your 90s, equivalent to Eric Topol's Welterly. So these are 80 and 90-year-olds that have never been sick a day in their life, never taken a drug, and so forth, that go into the 90s physically active and mentally capable. And of course, if you're willing to follow the scientific wellness, and this is a hypothesis that we're testing, but I'll show you why we think it's really going to be true in just a few moments. We started a company called Harel to be able to get 6,000 clients with their data clouds, and here I would never have gotten NIH to spend even $100 on this program, and we got venture capitalists to spend almost $70 million on this program. Arafat failed for really interesting reasons, but it was enormously successful in the things we cared about, and I'm going to show you just what those are. So 6,000 clients, and we saw more than 100 wellness to disease transitions, And that was for most chronic diseases. And we'll return to that in just a moment. So these data clouds, for the 108 and for the Harel, I think are really very much like the Hubble telescope that let us look at biology and disease with a different level of resolution than we've had before. And I'm going to give you, and I could show you a lot of data, but I'm just going to generalize it. I'm going to give you 5 examples of things that we've actually found. So the first, in the 108 individual, we found 3,500 beautiful statistical correlations, either positive or negative, between data bits in one of the 6 data types with data bits in the other type. And of course, what these generated was a very confusing hairball. But what we did was we clipped the edges that had the lowest probabilities, and these quickly brought into visualization more than 70 discrete analyte communities that correlated beautifully with physiology or with certain aspects of disease phenotype. And this gives you a powerful new statistical tool for looking at drug biomarkers and or potential drug target candidates and so forth. We confirmed that with the 6,000 individuals from Harel. And what was interesting is we ran the 108 people blood proteins against 5,000 human proteins. And we were able to move the statistical correlations from 3,500 to 35,000. And again, we saw systems associated we had no idea were operating in conjunction with one another. A second thing is we used polygenic risk scores. You can use genome-wide association studies to look at hundreds of thousands of patients that have various common diseases. And from that, you can derive tens to hundreds to even thousands of polygenic markers, each of which contribute a very small amount to the phenotype. And because we had complete genome sequence, we could take in this case, the 108 individuals, and we could determine whether they had very low, intermediate, high, or very high risks for, in this case, the expression of LDL cholesterol. And what was interesting, there were 3 things that were really interesting about these studies. Number one is when we ran them against all of the 1,200 analytes, there were a handful that had linear correlations beautifully like this. And this one is LDL cholesterol, which is not surprising because none of these people are on statins. And what happens with statins is you reduce the LDL cholesterol, and operationally you convert people from very high risk to medium or even low risk. And I'll just tell you without going into it, we've looked at 54 other diseases like this. We see exactly the same things. In some cases, we know why the analytes correlate. In other cases, we don't. But the other thing we demonstrated in Harel is if you have a very high genetic risk and you have very high cholesterol, lifestyle changes and exercise don't touch it. You need statins to change it. Whereas if you have the low risk and high cholesterol, lifestyle and exercise changes make a big difference. So you're going to be treated differently depending on whether you're high or low risk.
Yeah. This is not the ApoE4 or it's more complicated?
For ApoE4?
No, you said genetic risk. Is this the ApoE4?
No, that's completely a different thing. This is the correlation for LDL cholesterol and a tendency to have high levels of that. Completely different. Yes? Oh, okay. Okay, here's the third thing. What we found was with the 6,000 people, we could bend them into 10-year categories from 20 to 30 up to 80 to 90. And we could look at the expression levels or patterns for the three classes of analytes, clinical chemistries, proteins, and metabolites. And what we showed was a linear loss in the ability to control the expression levels of those things as you got older. And as you got older, you could see there were very light, broad bandwidths on individuals way above or way below those expression levels and so forth. And without going into any detail, let me say you can do calculations from these kind of data that allow us to calculate our biological age. And again, the age your body says you are. And the experiments we did were the following, really fascinating. So the idea is your biological age should be lower than your chronologic age, and the lower it is, the better off you are. So we looked at 300 people that had type 2 diabetes in this population. Their average biological age was 6 years older than their chronologic age. We looked at 250 people with cardiovascular disease. Their average age was 3 years older than their chronologic age. And we looked at those in the upper 5% of Fitbit exercise. Theirs was 3 years younger than their chronologic age. So you can ask, what happens to normal individuals? So my colleague Nathan Price and I actually had our ages analyzed all the way across. And what Nathan was able to demonstrate was, one, over the 4 years that he was in the Airvale program, he lost more than 5 years in chronologic age, so he's a total of 10 years younger. And he was an ideal subject because he's a Mormon, he doesn't drink, he doesn't smoke, he doesn't have any exposure to caffeine. And as I said, and the other thing that was interesting is the change, the decrease in biological age was beautifully linear across this 4-year period. So whatever was acting on it was acting in a stable fashion. And I had exactly the same data, only mine was a little bit younger. So my idea is in the future, we ought to have our biological ages done once a year, and then you can look back over that year and see whether you've aged well or not and make modifications accordingly. We saw 100 transitions from wellness to disease. The first was this woman who was diagnosed in January of 2017 with stage 4 pancreatic cancer. We had 3 blood draws roughly 6 months prior to, each 6 months prior to one another. And what we did was we analyzed for each of those blood draws individually the 1,200 analytes, asking if there were outliers. And at the time we did the experiment, we had 2,000 people in Airvale, and we beautifully saw one protein outlier that was statistically off scale. And it was in the Notch receptor pathway, a cancer pathway, and it was specifically expressed in beta cells of the pancreas. So it's a wonderful candidate as a biomarker or so forth. And I'm going to return to that, but what you can do with scientific wellness then is you can follow each individual continuously, looking for the earliest transitions for all diseases. But we'll come back to that in just a moment. And the final thing we did was show that we could look at 11 metabolites in the blood, and from those, calculate precisely the alpha diversity, the extent of diversity in the gut microbiome. And that's important because greater diversity is a good thing with regard to health. So returning to transitions again, our wellness studies are going to let us, for each individual, generate a baseline, and we'll be able to identify from changes in analytes and other things that we can follow, including disease-perturbed networks and so forth, when we have apparent transitions. And of course, the real question is, signal-to-noise issue and what's real and what's not real. And the key to is to get an enormous amount of data, and then we think in a 10-year period we'll be able to predict all of the major early transitions for most chronic diseases and be able to inform people that you've had this transition, and by that time, for many of the major chronic diseases, we'll be able to reverse them at that earliest state, and that prevents you from ever getting a chronic disease in the acute state if we can succeed in doing all of those things. And in the US anyway, 86% of the healthcare costs are chronic diseases. So if we can reverse that in a significant way, it's going to be major. And the one we've taken on first is Alzheimer's, because it really is a very complex disease. And I had a colleague who about 10 years ago asked a simple question: how can we optimize synaptic communication, one of the things lost in Alzheimer's? And he came up with a 36-point regimen that included drugs and supplements and lifestyle and sleep and a whole variety of related kinds of things like that. And he did a preliminary analysis with 100 individuals at the earliest stage of Alzheimer's and showed more than 80% of them could be reversed back to normal. If you changed back to your old lifestyle, you flipped right back to Alzheimer's very quickly. So if you're going to make the change, it means, again, you're going to have to make a lifestyle change. So what we've done then is, I'll just tell you in the last...
So what kind of change you need to do to reverse Alzheimer's?
I'll get to that in just... But it's all these things that I talked about. It's a whole series of supplements, drugs, lifestyle changes, and things like that, okay? So the history of Alzheimer's, you probably know, 400 clinical trials in the last 10 years, many of them north of $1 billion a trial, 0 success. And why has it been such a grim failure? And I think it's an appalling history. So what we've decided to do was put together the dense dynamic data clouds to study this process with this new multimodal therapy that I described just a moment ago, and together with cognitive brain health. And Mike Merzenich actually has pioneered over the past 30 years, both in animals and much more recently in clinical trials in humans, to show, one, you can use computer games to measure quantitatively your cognitive abilities, and he lists out about 25 of these different cognitive abilities. And number two, where you're deficient, there are games you can use to bring them back. So we decided we would use these cognitive games as a part of the therapy, as I'll show you in just a moment. We're establishing an Alzheimer's clinic close to ISB, and I'll show you why we want to do that. And we're going to be setting up 3 different clinical trials, as I'll show you. So the idea is the gold standard for when you get Alzheimer's is metabolic PET scanning, which shows changes 4 to 10 years before the clinical diagnoses are made. So we think that's where we want to go for our biomarkers. So we're going to use this deep phenotyping to be able to analyze these individuals and see the earliest transition. I'll say more about that in just a moment. We'll get the transitions, we'll get the earliest disease to wellness and wellness to disease transitions. And again, Alzheimer's has at least probably 3 to 5 different subtypes. We'll be able to get all of those. We're going to use the approach that was outlined in this book by Dale Bredesen for synaptic health and this multimodal adaptive therapy. Adaptive means you check the biochemistry of each individual and you treat them according to the deficiencies that they have. The cognitive health that we've talked about there was the other one. And the first trial that already we're a year into that's using cognitive health plus multimodal therapy and the deep phenotyping and so forth, the preliminary results really look promising. And one of the most striking observations is people that really went wild on the computer games for cognitive health really scored better than all of the rest of the people that did many of the other kinds of things. So we have two different clinics being set, one in Seattle and one at St. John's. And the first one, trial is already underway, and the second one, we're getting all ready to do it. The really key trial, which I haven't raised the money to do yet, is to follow this kind of strategy. So there is a website called ApoE4 Info that has 3,000 people, most of which are homozygous for ApoE4. They're at enormous risk when they get into their 60s for Alzheimer's. So we're going to take 1,000 of those individuals and initially test them with PET scanning so we can sort out those already converted from those that haven't converted. And we'll follow those that haven't converted with the dense data clouds to get the earliest possible transition points and markers so we can begin treating at this stage rather than when the disease has already manifested itself at the phenotype. So Providence St. Joseph's is big and has scale. It's 50 hospitals. It sees it's now 8 million patients a year and so forth. And it has 30 million electronic medical records that we can scan and so forth. We're setting up clinical trials using both longitudinal deep phenotyping I've talked about with systems-driven strategies, and the first three in dark there are clinical trials that are already underway. The scientific wellness trial has really had spectacular results in the Alzheimer's trial. The first one looks very good too. So, can I have a couple of more minutes, or I can quit now, either way?
Couple of minutes.
Okay. So the next big thing we're going to do is take 1 million people from Providence, and we're going to do genome sequencing, complete genome sequencing, and deep phenotyping. We'll have their blood in biobanks. We'll be able to get the electronic medical records. We'll be able to put all of that in the cloud, and we'll be able then to convert the cloud data into actionable possibilities that will transform how healthcare is treated. And I'll say more about that in just a few moments. To do it, it's going to be expensive, so we've already talked with many of these partners already, and we're getting lined up a set of people where we're going to have the technology companies and the pharma companies make substantial contributions to the cost of what is a very expensive program. One of the key things, of course, is to be able to convert the data in the cloud into actionable possibilities. And here, I think, is where we need to be able to create a knowledge platform. And we've actually hired a guy at ISB that has created a really interesting platform for Alzheimer's disease, taking Dale Bredesen's book, converting it into 811 concepts, each of which he calls an intelligent Lego that can interact with one another and interact with the patient's data as it comes in to be able to print out diagnosis and recommended therapeutic suggestions. But what is really key about creating that knowledge platform is the fact that individual patient data will come directly from Providence and clinical trials that we're doing there. But it will also come with domain expertise so that with each individual patient we put through this AI platform, we'll have the domain expert criticize it and modify it and improve it. And we figure within 200 patients or so, we'll have something that begins to approach the domain exercise. The other side, of course, is the N of 1 data will go to ISB, and we'll use machine learning to lead us to causal inference and hypotheses, followed by biological perturbations. And those conceptual building blocks will go in too. And we think you need all three of these things to really create the platform. I'll just say we're doing now exactly the same thing for the actionable possibilities from the genome and the phenome analyses of the million patients that we'll be talking about. The advantages are the longitudinal phenotyping. No one is really doing that now. The integration of the Airveil computational platforms with this very tentative initial AI platform for diagnosis, converting data to actionable possibilities, the partnerships. We're going to actually do a 5,000-person project starting first quarter of next year with normals and 4 different diseases, as you see there. And we're going to be bringing in other healthcare systems, both in the US and I'm actually talking with China about the possibilities too. So what is 21st century medicine going to achieve? 1, we're going to improve wellness for the individual and promote healthy aging. 2, we'll be able to identify the earliest transitions and hopefully reverse them. 3, this is really going to bring down the costs of healthcare. I haven't talked about that. And 4, wellness is going to migrate to the home. And I think these big capital efforts for a lot of healthcare systems are going to become more increasingly relevant in the future. So, just to give you an idea of what we're dealing with, time for medical knowledge to double. 50 years in 1950, 3.5 years in 2010, estimated 73 days in 2020. And however you do the calculations, they're going to be overwhelming. So we need a lot of help to figure out how we can handle this massive amount of data and convert it into actionable possibilities for physicians and their patients. So, thank you.
There's a clear separation between men and women, which traditionally in medicine hasn't been done properly. I assume that aging is quite different. I mean, and is that part of your study?
Yes, the Airveil population was slightly more women than men. I think it was 55-45 kind of thing. And the Benning that I showed you there was the mixed Benning, but we did Benning for women and Benning for men, and it wasn't substantially different, I have to say, by these. And it's pretty sloppy in those error bars, so it isn't going to be really sensitive. But we saw exactly the same kinds of things for women we did for the 2 men I described that were normal men. I will say we did do an experiment where we took all of the people that came into Airveil 5 years or greater, a biological age 5 years greater than their chronologic age. And for every year they stayed in, they lost a year of biological age. So that was it was the same kind of thing you saw for the 2 individuals I talked about.
Every year they lost 1 biological year?
Pardon?
For every year they lost 1 year?
1 year, approximately.
This can go on forever, essentially.
Well, I think you guys say nothing goes on forever, but I think we can get incredibly healthier than we are now at whatever stage we are. I will say that. That's the take-home lesson I took. It's a bit of work, but it's great.
Thanks for a very interesting talk. So at least in popular culture, there's this other sort of a stratum of wellness, which is all sorts of mental exercises, be it memory exercises, from memory exercises to meditation, socializing, having healthy relationships, and its actual effect on aging and biological age. So I was wondering, what's your take on that? Because it was kind of, I feel, left out mostly of the
Not really. I mean, what I certainly agree is socialization is really important for aging. No question about that. And one of the early things of P4 is network interactions. I didn't have time to talk about that, but I do think that is really very important.
Yes. Where do I sign up to get this list of things that I should be improving? Sorry, this was a joke, but it's actually a serious question. So you made a great list of things that affect you.
Well, that's for me.
I understand.
Yeah.
What I'm saying is, how close are we?
Come to Seattle. I've set a clinic up to do these kind of things. And actually, the clinic is going to be doing some other things I didn't talk about at all, including imaging. They're really inexpensive imaging tools that you can look at everything except the brain with.
Okay, February. I'll be there.
So if you come by, and I'll show you. Move to Seattle and we'll take care of you.
Okay, last question.
It's a continuation of Gal's question, and also a question
into the mic.
And also maybe a question that follows your talk from yesterday. So you mentioned these people who are coaches or guides who would tell the participants what they need to do. So do you think this is like some kind of new profession that would replace dietitians and family practitioners?
I've gone to a whole series of nursing schools, and I've actually persuaded one or two to really think seriously about doing this. I think the wellness coaches are really key because physicians don't have time to give individuals the time they need to do lifestyle changes. Physicians can't do, I remember when my physician said, well, you've got a family history of diabetes, we better watch that. And, he didn't do any tests to check it or anything like that, whereas the coaches are really hard nosed. What they were great at is they made you feel like they were your mother, and you felt guilty if you hadn't done the actionable possibilities you promised them. So.
Okay, let's thank Lee for